Thanks everyone for being here. And also before I start, I just want to say thank you to Margarita, Jean, Deborah, and also Roman for setting up that whole 4-day event or 4-afternoon event, and for having me as well. Yeah, I will be talking not that much about statistics, actually. I'm not coming from statistics. I'm not a philosopher of statistics, but I want to zoom out a bit more and reflect on the notion of questionable research practices, which is obviously within this debate as well. And it's just a broader issue that is being brought up. So to give you an overview of what I want to talk about, I'll give you my key claim first, which is that I think we should abandon the idea of a set of questionable research practices, or QRPs. The reason, as to clarify what I mean, is I don't mean to say that we should abandon the idea of questioning practice, but the idea that there is a class of questionable research practices. And I think there's an important distinction there. And to give you the reason why I think that we should do that is that, first of all, I think the idea of a class of QRPs is empirically inadequate, is a bad guide to a complex methodological landscape. And I think it's potentially damaging to science. And I think I echo also a lot of what David Hand will say, talk about in the next talk. Just quickly, what I think about QRPs, I think most people will be familiar with that, but I'll just quickly go through this. Obviously, this idea of QRPs, and actually Daniele, who just spoke, has done a lot of very important work, I think, as well, and I'll reference him later on this. But the idea of QRPs has been obviously very dominant in the debates about scientific integrity for about 30 years or more, but also in the more recent debate about replication crisis. And there's often this quite admittedly very neat, seductive, linear causal narrative, this little mechanism. On the right-hand side, we have a flawed scientific output, something's going wrong, or we can trace that back to maybe an increased use of QRPs. And that increased use we can trace back to some sort of flawed incentive structures. And you immediately get kind of a recipe for intervention. So you get, we'll improve our incentive structures, we'll somehow engineer them. That will lead to a decrease in use of QRPs, and it will improve our flawed scientific output. Sounds very straightforward and nice. To give you an example of just one paper, there's different authors who make this move. I would say it's quite common, but just to give you one example, Glenn, Bagley, and Ioannidis claim that the replicability crisis is not due to a failure of the scientific method. So that's always kind of protected by a lot of people who talk about this problem. But they say it's a consequence of a system that is willing to overlook and ignore a lack of scientific rigor on behalf of the scientists and instead reward flashy results. And then they say also, although fraud is rare, the use of questionable research practices seem to affect the majority of researchers. So we can find a lot of QRPs and, admittedly relatively low levels of actual full-on fraud. And why do we have this problem? That goes back to this kind of distal reason, because they say we get what we incentivize. Yeah. And so there's several papers that talk about this also in philosophy of science and trying to model how this works, where the key incentive is this kind of prestige or credit. And so you build your prestige through publishing new results, and the QRPs are usually represented as a shortcut to get more results, to get them faster, and to get kind of higher impact or sexier results. And so you get this linear narrative slightly updated. So the increased use of QRPs is because we need more output, and we need more output because of the credit economy. And so, yeah, you can see that obviously QRPs are really kind of a pivot in here in the middle. And sometimes it's not, it's not that unicausal kind of the monocausal model. Like, it's also often highlighted, there's a lack of knowledge of proper method. And kind of, these are both, I'm not going to say these are not factors in the equation. Obviously, we need to have, education. Everybody learns scientific methods in some way, but what I just note here is like there's a very one-dimensional view here of researchers, and the kind of, I come from biochemistry originally and moved into philosophy of biology, and it just, I don't know, it just touches me personally. When I read these things, it's kind of, well, the researchers are either greedy or they're kind of dishonest, two going together presumably, and then they're a bit stupid or don't care. And so as a result, you have to change the incentive structure to engineer their behavior, kind of nudging, paternalism, whatever. You have to increase surveillance or control, and you have to enforce more training. And so, yeah, anyway, that's kind of what comes out of that. Obviously, in this whole scheme, the question is like, what are these QRPs? And you have basically the term, I guess, started being used in the 1950s, 60s, but it really came to more prominence, I think, in a National Academy of Science report in 1992 on responsible science. And here it was defined as questionable research practices are actions that violate traditional values of the research enterprise and that may be detrimental to the research process. Then what I have in brackets there is a little section on saying that actually there's no consensus on what they are or what the standards would be. And then they explicitly say that QRPs do not directly damage the integrity of the research process. And I think that was 30 years ago. They gave some examples. This is not all of them, but for instance, one is you would fail to store data for long enough. You would exploit your subordinates, like PhD students or technicians. You wouldn't share unique research materials with other researchers, or you would kind of ask to be on a paper, even though you haven't really directly contributed to results, but maybe just, gave some specialized service and like, yeah, put me on the paper. Anyway, so these are kind of from 30 years ago. There was also misuse of statistical methods. That was the only one that will come up later again. But then about 20 years later, in 2012, when the whole replication crisis narrative came up, the QRPs became explicitly connected to the idea of the crisis. And there's a couple of papers that made this move. And the idea here was that, these practices are actually detrimental. They're used to massage and torture data and so on. And actually, the National Academies of Sciences have a 2017 report on integrity and reproducibility and responsibility. And there they actually, go in and say it should actually be DRPs, detrimental research practices, not QRPs. So there's really a shift in there of what they are. And now the examples are more kind of the whole p-hacking, hypothesizing after results are known, harking, or lack of experimental controls and other factors. And I think a second thing that happens there is just that whole narrative basically becomes supercharged, because now it's also QRPs, or the use of QRPs is now seen as a sign of dishonest science. And I give just two examples here, Dan Sarevitz, or also Remco Heesen, who models kind of the relation between incentive structures and QRPs, or reliability of output. There's like a narrative of like the honest scientist who doesn't use QRPs, and then, by implication or maybe explicitly, the dishonest or fraudulent scientist who uses QRPs. So the narrative is really ramped up. And it becomes a proper tool, obviously, to inform and guide the debate about policy interventions. And in many ways, it's kind of, or at least that's often how it's used, as a kind of a neat distinction, and it promises neat solutions. So you get this kind of this picture now with just kind of three parts of science. You have the GRP, or good research practice. Some people talk about responsible conduct of research, RCR. Anyway, it doesn't really matter. In the middle, you have the gray area of QRPs. And then in red, you have FFP, falsification, fabrication, and plagiarism. This is the really bad stuff. But you can see at the bottom, like, what's dishonest science is quite expanded. The solution is quite obvious. You need to somehow, we're trying to get rid of QRPs. And I guess there are two ways of how, or they're related, how you can do that. If you have 6 different QRPs, you just try to reduce the use of each of them. And by that, you bring down the whole level of QRPing in research, or you try to eliminate specific ones and just kind of forbid them altogether, these practices. Or, I mean, elimination can be an extreme case of reduction. You just bring it almost to zero or to zero. And that brings us back to kind of, well, if we do that, then what should we target? And so in the 1992 discussion list of the NAS report, we saw, some suggestions for QRPs. As I just said, in the newer debate, it's kind of shifted a bit. And if we look at the debate now, after 30 years of quite some research on this, we still don't have like the consensus on what this list of QRPs should be. So Lex Bouter and his colleagues, they had a big study. They produced a list of 60 QRPs. Raven and Sørensen, I'll come back to that paper. They did focus groups and interviews with scientists. They did 105 scientists and they got 107 QRPs. It's like, you ask 10 people and you get 12 answers to what quantum mechanics is, or, it's really kind of this almost. And then they reduced it down to 34 by like coding and elimination of duplicates or whatever, however they've done that. But it's obviously described in the paper, but like they bring it down to 34, which doesn't really match the list of 60 and so on. So you can see the point. You could say here, obviously, well, yeah, I mean, any classification will have some problems at the boundaries. I mean, this is the wrong way to look at it. You could say, as long as we have some clear examples in the center, surely we can deal with, this is just part of it. Everything is messy, so we'll get some vagueness. So, we have these well-established examples that come up again and again. Lack of controls is one that's often mentioned. Glenn Begley uses that as one of the 6 red flags of suspicious data. And then obviously p-hacking, harking, publication bias, and so on, or other kind of questionable practices. And so you could say, well, let's just focus on these. Yeah, this is what we need to eliminate or reduce. And this is really where I want to kind of look a bit closer. The first key kind of QRP that I want to look at is a lack of experimental controls. And I'm speaking here of kind of biomedical research, and you have your experimental setup, animal study, or anything where you need your negative and positive controls to actually say whether something is an artifact, false positive, false negative. So same kind of type 1, type 2 error debates that you have. You just want to exclude false positives. And the idea here obviously is, well, and it makes sense at first sight, we need to enforce that only experiments with appropriate controls are produced and published. And I think Begley, I just pick up on Begley and Ioannidis here because they make some suggestions on how to re-engineer the choice architecture within which scientists work. And they talk about compulsory refresher courses on experimental design for all researchers. I don't know who will teach those or on what kind of textbook, but anyway. Another one is to monitor, investigate the compliance with peer-generated guidelines and reward them. Like, don't know what the reward is, whether it's financial or a pat on the back, but that's kind of obviously it will be more sophisticated, but you get the language here. And then there's also obviously like change in editorial practice. You ask for the right controls to be taken, which I think is already practiced, but maybe, enforce that more. And so the idea here is really to enforce a more standardized landscape of controlling practices. And I guess that, sounds innocent enough and it makes sense. We need negative and positive controls. I just want to bring in here one example. And so I need to do a little bit of biology or biological methodology. I hope it becomes clear enough for the point I want to make. This is one particular method here. It's called RNA interference. Basically what you do, in case you can see my pointer here, you have what are called short interfering RNAs. So they have usually 21 nucleotides, and you can design them. You give them a specific sequence. And within your replication, within the cell, you add them to cells. They become kind of single-stranded RNA bits. And because you design them, you can design them so that they're complementary to a specific messenger RNA within your cell. And so the short interfering RNA will bind to it very specifically, only to that particular type of messenger RNA. And when that binding event happens, it triggers a cascade of reactions, which brings like a complex of proteins, which is kind of this yellow thing here, which leads to the cutting of this messenger RNA, which you can see down here. And once that happens, it's not functional anymore. And that's relevant because the messenger RNA is used to produce protein. So suddenly that specific mRNA is not present and that protein is not produced anymore. So over a day or two, because the cell has a natural turnover, that protein will start to disappear because it's not reproduced, not produced anymore. So what you can do is basically you can create mutant cells that lack a particular factor, and you can run experiments. How does the phenotype change? So you don't have to make actual genetic mutations, but it's usually called knockdown because you knock down a particular protein. Anyway, and so a very powerful method, and you do like whole screens, like with the whole library of siRNAs. Anyway, so what you obviously need to do is you are adding a particular RNA here, component, and so you want to know that just adding this on its own doesn't cause any kind of effects, because that would just be an artifact. You would say, oh, something has happened, but actually it's just because of the intervention you do, not because of this particular event here. And so what people did was they basically took this sequence, the same 21 nucleotides, but just scrambled it. So it wouldn't recognize anything. It just becomes a nonsense sequence. But what you have is that you still have the same chemical composition. So you get as close as possible to the actual intervention, and you have like a very good negative control where nothing should happen, or just unspecific background. And the idea would be whatever scrambled sequence you have, because they're nonsense, they just will give like the same kind of background presence of nonsense 21 nucleotide RNA. But what actually happened once people started looking more closely, and we used that for years, people started looking a bit more closely and they realized, no, actually different scrambled siRNAs actually upregulate some genes, downregulate others. It's each has its own specific effect. So you can really not use it as a negative control because it does something quite specific, and another scramble does something else specific. So it becomes an intervention on its own. So people started actually leaving out these negative controls because they're not negative. And you can see here, I mean, this is just one example, but I think it does imply that actually in this case, the use of the control would be questionable, not the lack of a control. And so if you start enforcing this kind of, well, any experiment that lacks negative controls should not be publishable, could be detrimental in some ways, in some contexts. So I think there can be no fixed classification here. What counts as a QRP in one experiment in the same field, discipline, is actually a GRP in another experimental context. And so you have contextual instability of this classification within disciplines. Another example here, and this is what I had in my abstract mostly, but I try to tone it down because it would just be too much biology and kind of not really get to the point as quickly. Is another example is a far Western blot. And I'm not going to explain the details, but just the basics is it's basically used to study the interaction between two proteins, protein A and protein B. And it's a very simple setup. So it's very powerful, easy, cheap to use, and you can use it quickly and so on. But it has a huge problem because proteins are usually structured and then they interact. But to use this particular method, you need to completely destroy the fold of the proteins. And that means you need to refold them first again. So it's a methodological step. But this refolding is really difficult to achieve. So most often you have like only partially refolded proteins, or they remain unfolded and you don't really know. And so you will have a lot of non-specific binding events because you still have unfolded protein present. So you get a lot of false positives, like, oh, it binds. Well, yeah, just because you unfolded it and kind of. So it's a method that when I was still in biochemistry, we would like shoot down at the review stage, we would shoot down any paper. If we read just FWB, we were like, no, we were highly skeptical and was really shunned by a lot of people. So it was treated as this is a questionable research practice because it generates so many false positives. should not be used. Now, in the 1990s and 2000s, and that didn't become dominant until the 2010s, I would say, people started discovering proteins that actually are never folded or never fully folded. And they're called intrinsically disordered proteins. And this has really changed the paradigm thinking about proteins and so on. And it's a significant amount of proteins. It's not just like, 1% or whatever. It's like quite a big part. And in many ways, that rehabilitates the far Western blot, because for a protein that never folds, the whole unfolding, refolding problem is just not relevant. And so you can say that this assay probably works beautifully for a significant class of proteins. And some people, were always like, who worked with the method, were like really believing it, and like we got great results with it, and like everybody else was like, it's just like you just don't know what you're doing, really, or you're lucky and you have like that one protein for which it works. But actually, they might have had good reason, and it might have been a very powerful method to assay at least IDPs, intrinsically disordered proteins. And so anyway, here again, I think what counted as a QRP turned into a potential GRP, and I think there's also there's a diachronic instability of this classification within even one discipline. Okay, and this brings me back to the Raven and Sørensen study. Daniel Lakens, can I just ask you quickly how much time I have left?
You started a bit late, so if you want to use 3 extra minutes in total, that's fine.
So how many have I? So when should I answer?
8 minutes.
Oh, okay, great. Yeah, I'll be fine. Thank you. So I'll come back to the Raven and Sørensen study. So what they did is they did these focus groups across different disciplines, humanities, natural sciences, engineering, and so on. And even though they didn't find a consensus with other studies like the Bouty study or others, but what they pointed out is actually the diversity across different cultures, which might to some sound common sense, but it's something that had a point that had to be made specifically in 2021, and is like, I think is a relevant novel contribution. And so they say what scientists count as a QRP in one discipline is seen as a GRP in other disciplines, or even within one research area. So they say they work with Karin, Knorr, Cettina's epistemic cultures view. And so they say there's not only is there no unity in the creation and justification of science, there is neither a unity in QRPs. And so they point again to an instability of classification within and across individual research areas as a result of their survey of scientists. This point here, I'm going to make it relatively short because I'm not necessarily a specialist, but other people, also some who are present here, have done work on this. You could obviously say, yes, Stephan, that's all great, like your controls and all that for Western Blot, fine. But like, what about the classics that we like to talk about, like p-hacking, harking, and all this? And again, I'm not a specialist on this, but there are studies who as far as I can assess it, show quite nicely that, for instance, p-hacking does not necessarily damage replicability, or it might not be a major contributor to low replicability. Rubin showed or argues that it's premature to think that harking, hypothesizing after results are known, is contributing to the low replication rates that we observe. And Daniele, obviously, who just spoke in his PNAS lovely paper, 2018, argues that the QRPs are present if this is what we accept as the definition of QRPs. But he says that they do not distort the overall scientific output, or not distort it significantly, at least at current levels at which they're supposed to be present. Alexander Bird, who I'm not sure whether he's here, but he also has this lovely paper where he says, well, actually, low replicability is compatible with high quality research, because actually it's a high base rate of false hypotheses. That's the key problem, not necessarily a lack of quality in research. And Ulrich and Miller, I think, trying to make the same or similar point. So, I mean, when we come to these, I think, again, there's just no general statement about the goodness or badness of these practices even. So not just p-value, but even p-hacking. Some people have their questions whether it's that damaging in the end. So to kind of summarize bring this together, I think when we, like the question here, are there questionable QRPs, or is the assignment of the label QRP very questionable? I think even for those QRPs on which there is some consensus in the existing literature that these are the QRPs, I think there's diachronic instability that former QRPs become classified as GRPs or vice versa. There's synchronic instability. You have within or across different disciplines, one can be a QRP or a GRP. And you also have, I don't know whether that's the right word, but like causal inertness, you have the presence, but they don't really seem to make a difference or that have a detrimental effect, which goes straight against what the National Academies of Science report in 2017 says, which again was hypercharged by the replication crisis debate, where they said like, yeah, we thought we could treat them as QRPs, but actually we know now they're detrimental. They have a direct impact. But I think Daniele's studies and others show that maybe not that good if you look at the evidence closely. So to summarize, I think it's very hard to point to individual practices that are inside or outside of this class of QRPs if we have this tripartite distinction, GRP, QRP, fraud. And so I think the label does not serve as a good guide to a complex and shifting landscape of experimental practice. The second point, I think the label is used in a universalizing manner, and it has become supercharged as, the detrimental practices. And so it does encourage blanket exclusions of certain practices. It encourages the formation of a homogenized methodological landscape. And so I think what we have here is we have a label that's an inaccurate guide, but at the same time yields great normative power. And so I think that's a potentially very damaging situation. And I think we might do better if we abandon talk of QRPs. I'll just bring that quickly back to the NAS report. They were worried about governmental influence. But anyway, what they said is like governmental or regulatory efforts to define correct research conduct or analytical practices can do fundamental harm to research activities if such efforts encourage orthodoxy and rigidity and inhibit novel or creative research practices. And I think that warning still stands, or it speaks to me at least. And I think various people also in the research integrity research, so Valkenburg et al., for instance, they look at research integrity. They are trying to promote an approach to integrity that focuses more on local values and epistemic cultures, focuses less, well, first of all, abandons universalizing tendencies, look at local limitations and potential of different practices or cultures. So you can still question stuff, but just in a different manner. It would entail stop resorting to this kind of moral panic about the dishonest research and how they use QRPs. Alexander Burr makes a similar point, like with his explanation of where the replication crisis might come from, if it's a crisis. And then generally that would entail, if we start looking at values and epistemic cultures, that we mobilize a more pluralistic approach to tackle the issues of questionable research. So that would be empirical, something that Daniele is trying to do also with his K theory, I guess, to sharpen that, but also conceptual and historical, sociological, philosophical. So that will be the three things. And I think I'm going to stop here. I have two more slides. That just somehow this QRP talk goes very broad. Like some people attack animal model research, like Sorowitz, as kind of questionable, or people kind of, a non-applied basic research should be reduced and so on. So it goes off on quite a thing. But anyway, I leave that out here. And I'm just going to say thank you for your attention. And I don't have the references in. I didn't have time to put them all in, but you can email me and I can send you the papers if you don't know what it is. And I'm going to stop sharing, I think.